All right, so good morning, everybody. I just uploaded the next PowerPoint after the one we hopefully finish today. I'll put the reading assignment, but it's basically chapter 2 and a little bit of chapter 4 as it relates to archaea. You'll see eukaryotes isn't a lot, but right before I started recording, I bumped our test back to Monday, September 12th. I was being ambitious in the beginning. I don't know why with a new book. I don't know why. I was also having trouble judging because I haven't done a 50-minute class in a long time. I'm usually doing hour 15. So this new PowerPoint actually has the new pictures from the new book, right? Real pretty pictures. So there's a couple of features in the book that I wanted to point out to you guys. Like, for instance, like I said, and look, I guessed correctly. Mainly this topic is going to be covered in Chapter 2, which is bacteria, but we're going to refer to some components of archaea, which are also prokaryotes, in Chapter 4. Chapter 4 has this really cool graphic, and it has a podcast that you can click on, and they'll tell you about this image. And so when we go to our modules, the other thing is we've definitely finished the first topic. So I have the first Wiley plus. I was going to say the other word. I was pausing, trying to find the right word. The first Wiley plus assignment right here, due for Wednesday. It's 14 questions, 14 points. And then I've already put together the one for the next lecture. And then there'll be one more for the last topic. I'm not going to do one for microscopy. I kind of included a question or two from this lecture in prokaryotes.
Question about Wiley. When you're doing Wiley, do you get one shot at it, or do you
No, I said my homework says unlimited, but good question. One of the things I suggest students do is you study that topic, right? Then do the assignment as if it was a quiz. Close your notes, close your book. See how much you actually know. Because you can do it again. It's not going to hurt you to do it that way. It's only going to help you. Actually figure out what you truly know and don't know before a high stakes test. That's not when you want to find out whether you know something or not, right? So I'm not going to be there to babysit you. You guys do these on your own. But that's going to be your best course of action, right? So I have a due date set, but you can do it after the due date, and it's not going to take points off. But I want you to do it at least once before the due date. Make sense? Right? So these are guidelines. So I got distracted. Oh, I wanted to go to the book. So read, study, practice. Load in a new tab. I was playing one of the animations on my phone. You guys know about the Canvas app, right? So for Canvas, and Wiley works with it, right? You click this link, it'll go right to Wiley. The animations played great on my iPhone. I have an iPhone. I don't know about Android, but it should work. They work? Awesome. So I go to chapter 4 and the illustration podcast right there. But I think if you click chapter opener, it's there too. So there's the picture and there's the podcast. What I'm concerned about why I'm going to this is, unfortunately, I don't see the little caption signal for Vanessa. This is not required content. It's kind of nice. So I'm surprised that they don't. Oh, look, download script. I take it back. There it is. So there you go. It's a Word file. You just open it up, and there's the script. I was going to say, it's not like them to not be ADA compliant. I just didn't remember seeing it. So the other thing I want to point out that I absolutely love and listen to about a gazillion times including, again, just right now, especially for Chapter 2, if you don't even want to read the book before Wednesday class, please just come to it and at least go to the animations, the chapter animations, these 3D ones. Watch, there's 4 of them. 1, 2, 3, 4. Watch them at least. If you do nothing else, and they will play on your phone. I was playing it on my phone, right? And they are closed captioned. So just turn it on. Okay? All right. They are excellent. I suggest some of these other ones too, but these are the core, especially for Chapter 2. So you'll see that in my PowerPoint too, if you download this and you put it in presentation mode, I link them in, but you have to be, notice, connected to the internet. So then you click on it and it goes to the internet. Right, and you can, I think I have the captioning on.
Are smaller than eukaryote cells, but significantly larger than most viruses.
Why isn't it showing up?
Some bacteria are
There we
extremely
go.
small
I had to select the
compared to other
language.
bacteria,
Apparently it does
and
more
they're
than
referred
one.
to as nanobacteria.
So, they're great. Beautiful. Alright, so I'm going to leave the internet open. Close out this one. So I just posted this one. It's long. It's going to take us about 2.5 days to go through, I predict. We'll see how it goes. Alright, so where we are at is still microscopy. We were talking about resolution and how numerical aperture comes into play with that. So that ability of the lens to focus the light into a nice, crisp, clear image. And so that is set for each lens. Its magnification is set by the X, right? So this magnifies 10X, and it has a 0.4 numerical aperture. And that could be used to calculate the resolution or the resolving power of that particular lens. So the other problem we run into is the refraction of light as it moves through different media, because light is a wave and it travels at different speeds through different media, and therefore it affects when it gets to our eyes and the image that we see. So this pencil looks broken, right? But we know it's not. This is that optical effect of refraction, where the light passing through the water as opposed through the air is moving at a different speed. So therefore the image is not connected. This affects fishermen too. If you're, say, trying to spear fish out of the water, the fish will appear at a different spot than it actually is. Right? You're best to get in the water, be in the same medium as the fish to be able to see where it's at. So I don't know, when I was a little kid, we used to try and scoop minnows out of the creek at my grandmother's house. And, you know, the only reason we caught any because there were like 20 of them all in the same area at the same time. If we were trying to catch individual fish, that would be difficult. But if you watch bears, right, and they just scoop the fish up out of the water, you kind of have to adjust for that. Then, of course, it's moving. It's a whole nother level of complexity. So do we deal with this when we use microscopes? Refraction of light? Definitely. So if you're in lab, you've learned a new technique in microbiology lab that you've never probably used in any of your other labs that use a microscope. And that is the use of oil. Because at 1,000 times magnified, which we can do with the oil objective lens, which is 100 times, and our oculars, which are 10 times magnified, that lens gets really close to the slide. And because it's going from glass and then into air, the speed is going to be different. There's more refraction of that light away from the lens. And you need that light focused into the lens so you can actually see it. So to help with that refractive index problem, we add oil to the slide. Just a drop. And that oil has a similar refractive index to the glass. So the light keeps heading at the same speed and hopefully straight into the lens. And this is why in any of the other classes, if you went to oil immersion, you never could get it focused, because most of the light was being refracted. Adding that drop of oil makes an immense difference. Most of you guys have already experienced that. And the oil, the nice thing about it, it's flexible, right? So you can focus up and down and still focus all that light. Does that make sense to you guys? Right. If you don't clean that off the lens, eventually over time it creeps up into the apparatus and gets behind the lens and makes that lens permanently blurry. So when we're done with our microscopes for the day, we always take lens paper and wipe this oil off. In the microbiology lab, most of our high dries are permanently blurry for that reason. For students using oil when they're not supposed to, high dry means dry, and not cleaning up after themselves, which is unfortunate. Bacteria, though, best viewed under oil, and that's primarily what we do in microbiology lab. So different microscopes are going to give you different abilities. They show you lots of different ones. One of the things I like about the new book is that you'll notice all the images have a little box if they were taken using a microscope, and most are when you're in microbiology. LM stands for, anyone know? Light microscope, and particularly a particular type of light microscope that we don't have here at Delgado currently is a phase contrast microscope. And the cool thing about these is they use special filters that help refract the light on purpose to show subtle differences between the organisms in the background. And the great thing about this is we don't have to create contrast with staining like we normally do. And some microscopes, like the difference inference contrast one, this amoeba looks like it's about ready to attack you, huh? It almost even looks three-dimensional. So you can see really thick specimens that you wouldn't normally see clearly under the light microscope, standard light microscope that we have in lab. So why would you choose to use this type of microscope as opposed to the ones we have in lab? Why do we have all these different types of microscopes? Huh? The organism's too small? It's a light microscope, so that's not going to help really with something that's small. Amoeba are really big. You guys can see them in lab, right? Can you see the whole thing at the same time, though? No, not really, depending on how thick it is and the magnification you're at. It really depends on what you need to see, right? Will determine on what microscope you're going to do. What type of research are you doing? What type of analysis are you doing? That's going to determine what microscope, because different microscopes have different abilities. You're not going to try and look at a virus with a light microscope, right? You're not going to see it. So if you're studying viruses, you're using the more high-powered microscopes. Or maybe if you're even studying stuff at the molecular level, but you're using the aid of fluorescent dyes, like you see here in these pictures. This is still light microscopy, but a special kind. FM stands for fluorescence microscopy, where they've been stained with fluorescent dyes or the organisms are fluorescent. You're using UV light and special filters to be able to visualize these different dyes. This is becoming more and more popular in research, to be able to visualize things, specific components. So there you can see the individual protein fibers within this cell because they've been labeled with a green fluorescent dye. But again, if you're going to look at things like viruses, or you want to really see in detail the inside of a bacterial cell, then you're going to need to use an electron microscope. And there are two different types of electron microscopy. There's transmission electron microscopy, where the specimens are cut into really thin pieces. Or, and again, notice TEM stands for transmission electron microscopy. So as you see these images throughout your book, you know what this stands for. And then if you see SEM, this stands for scanning electron microscopy. And as you can see, we're just kind of scanning the surface. So we're just seeing the surface. But you can learn a lot about an organism by seeing its surface. The images that are generated by the machine that's utilized for this, we can't see this with our eyes, are grayscale images. So typically they look like this. But a lot of times what they do for visual effect is they color it. So it's like taking an old-time black and white photo and literally coloring it. So they do that a lot with electron micrographs, images from electron microscopes. So the same was done here. So notice this one's grayscale, but this one they colored. So this microscope's really cool. I've actually had an opportunity to watch someone use one of these. It uses lasers. It takes several images slicing through the specimen, and the computer can generate three-dimensional renderings. And again, if you use fluorescent dyes, you can see different components. So they did that for this picture too, that they talk about in Chapter 2. Being able to visualize these structures that they believe to be an organelle. And this is a prokaryote. So it's like, uh-oh, our definition's got to be adjusted a little, right? But they're the only ones so far that they've discovered that actually has membrane-bound structures within the cell that are organized to what we would consider an organelle level that you see in what type of cells? What cells typically have organelles? Eukaryotes. But this is a prokaryote. By definition, they don't usually have organelles, right? Their definition means without a nucleus, which is an organelle. These are the really cool microscopes that I haven't seen yet, the more advanced ones where we can see down to the atomic level. The scanning, tunneling ones that use atomic force. And again, this image has been colored to help visualize the different parts. So this is actually DNA. You see the double helix? Right? It's approximately 3 turns. These are aquaporins, which are specialized protein structures in cell membranes that control the flow of water in and out of cells. Dr. James Gunther will tell you a whole bunch of cool stuff about these if you love proteins. He loves proteins. He's the director of our biotech program here at Delgado. So as I said earlier, pretty much to create the contrast we need to visualize the organisms, we're going to stain them. And there's lots of different types of staining procedures that help us visualize specific structures. Simple stains, on the other hand, are just that. They're simple. They're not going to tell us a lot, but they are going to allow us to see the organism. Gram stain, acid fast, and endospore, these are all specialized stains commonly grouped into a definition known as differential stains, where different organisms are going to stain differently. The capsule and flagella stains, as the name implies, are specific for those structures. Right? So those stains we do specifically to see and be able to better visualize a capsule or a flagella if they're present. So what's a simple stain? Using a single dye. Why do we stain them?
Like one color.
One color, one dye. Everything's one color. It's monochromatic.
See, right?
Yeah, and that's why we stain, so we can see, right? So we can see them. So as you can see in these pictures, we can see something, right? We can see these purplish blue cells. We can see these blue cells. We can see this gray background in these clear cells. There are two main types of chemical dyes that we use. Most of the ones we use are called basic dyes, and then we use some acidic dyes. Basic dyes have a positive charge, and therefore they're attracted to what charge? Negative. Opposites attract. Explains my first marriage. Okay, so what charge do you think cells have? They're negative. Their membrane, those phospholipids that we'll talk about, right, are highly negatively charged. And then DNA inside the cell is extremely negatively charged. So they're attracted to the membrane, they're attracted inside the cell. So basic dyes will stain the cell. So that's what you see here. Crystal violet, methylene blue, these are basic dyes. They stain the cell. Acidic dyes, therefore, are the opposite, right? They're negatively charged. And what do you know about like charges? They repel each other. It's like trying to stick two magnets together the wrong way, right? If you have the two same poles, they just keep flying apart, right? You got to flip it over so that the opposite charges are interacting in order to get the magnets to stick together. So the same phenomenon happens here. So instead of the cells staining, they're repelling the dye and the background gets stained. So what can you actually tell me about these bacteria? We can see them, right? We can describe their shape. You can see how there's even like two of them grouped together right here. That's about it. What we call morphology, right? Shape, arrangement. Sometimes that can be really beneficial, especially if you have a Staphylococcus aureus infection. They cluster. They're cocci shaped. That'll give us a good clue as like, oh yeah, maybe it is a staph infection. When we do these differential stains, this is going to tell us a little bit more information depending on the stain. So differential means we're going to have differences, right? We can group organisms based on these differences. So this allows us to still see morphology, right? You still see the shape of these cells? What type of dyes do you think these are? Because they're staining the cell, aren't they? Are they basic or acidic? They're basic, right? So we're staining the cells, but what do you notice in this picture? What colors do you see? Purple and pink. We've got two different colors. We've got two different cell types. Specifically, their cell walls differ for gram staining. Acid-fast, their cell wall components are different too. You have two different colors that are kind of hard to see here, right? You have the fuchsia, purple, pink color all kind of clumped together, but notice all the little blue? That's the other cell type, the non-acid-fast. And then this one happens to have endospores. So the endospores are these little pink things. The cells themselves are kind of this bluish color. Those of you guys in lab will do all these stains.
What was the pink?
The pink are the endospores, because this is an endospore stain. So for acid-fast staining, we do this to detect a particular group of bacteria called mycobacteria. So I mentioned a mycobacteria previously. It causes a disease that we grow the organism in armadillos because we can't grow it in petri dishes. Leprosy. Mycobacteria leprae is acid-fast, right? It has mycolic acid in its cell wall, so it stains with this staining procedure, this pretty pink, fuchsia, purple color. All the rest of the bacteria pretty much stain non-acid-fast. But if we're dealing with this type of organism, this type of pathogen, whether it be Mycobacteria leprae or Mycobacteria tuberculosis, this would be really beneficial, right? To do a quick stain and know, oh, hey, We're dealing with this genus of bacteria. We need to figure out which species now to be able to treat our patients. So we literally could get a preliminary diagnosis just by staining using the acid-fast stain. Endospores too, right? Knowing that an organism has endospores can be really beneficial in identifying it. And then, as these guys say, right, what do these stains allow us to see? Well, a capsule stain allows us to see a capsule. A flagella stain allows us to see the flagella, right? Why do you need special stains to see these structures? So when you look at the capsule, so here's the cell, it's only partly stained. This clear halo is the capsule, and then you have this dark background that's also stained. Capsules don't stain. So when we do this in lab, it's actually a two-step procedure. We stain the background, then we stain the cells. And what's not going to get stained is the capsule, if it's there. So that's why we've got to use this special technique, because you're not going to be able to visualize these otherwise. The flagella. We have to do a special stain. Bacteria are teeny tiny, right? They themselves are barely within the limit of resolution of a light microscope. Those of you guys that are in a lab in the next couple of weeks, you're going to be like, oh my God, they're so small. And we're at oil immersion. A flagella is a little tiny hair-like, right, protein appendage that sticks off the bacteria. So if the bacteria is teeny tiny, you're going to see some little tiny little hairs coming off of it? No. No. So instead, what this stain does is it adheres to those specific proteins that make up the flagella, and they make it bigger. It's like mascara for bacteria flagella. Makes them thicker and bigger, right? Guys, you still know what I'm talking about, right? What mascara does. Okay. Makes the eyelashes look longer and thicker by applying black goo to them, basically. By doing that, that makes them thick enough that you can actually see them with the light microscope. We stained them, didn't we? Are they still alive? No. When you stain, you kill. So when he was alive, he moved. He seriously moved. Not moving anymore. He's in its suspended animation. But he's covered in flagella. Like, you know, cousin It from the Addams Family. Just covered. This is an organism we used to work with in lab, Proteus vulgaris. It was one of my favorites. But it's a pathogen, so we got rid of it. And you can guess by its name, how did it smell? Gross. Proteus vulgaris, yeah, it stunk really bad. So you may ask, why was it one of my favorite? Because it smelled bad. And it was easy to grow. Me and him were, you know, we were all good about growing for me and not protesting. Because if the organisms don't grow, you guys don't have anything to play with, and I'm in here on the weekend. So, for gram staining, which is a differential stain, right, we're going to end up with purple and pink. We've studied this quite extensively, and we have a pretty good idea of why you get the different colors, the purple or the pink, and how it relates to the cell wall structure in bacteria. So there's an animation from our old textbook that, of course, everybody has put on YouTube. So it's going to time me out. I have it under modules. And I had put right here for you guys to read it. I mean, watch it. So notice right here, watch, right? So then it takes you to that other page, which is in that module, and here it is right here. I highly suggest watching it. I'm not going to play it in class. It does have captioning, correct captioning, because here, I'll open it so you can see.
The Gram stain is the most widely employed staining method in microbiology.
They cut out the captioning
It
on
is
this
a
one.
differential stain because it divides bacteria into two classes.
But it does look like it's
Gram
correct.
negative. In the first
So,
step
that's
of
good.
the Gram stain procedure, cells
So
from
let's
a
pause that one. This one does not have any audio, so it is definitely captioned. There's no audio. I'm going to kind of add my own audio right now. And this is my professional one that the audio-visual department did of me actually doing the Gram stain. So those of you guys in lab will really like this one. And of course, it's on my lab site as well. These are other beneficial ones that are helpful in addition to the ones that your book has, which are awesome. So I'm going to go ahead and link out to this one. And thank you to these wonderful professors at North Carolina State for putting this together, because it is awesome. So we have two outcomes, gram positive or gram negative for the Gram stain. This positive and negative doesn't have anything to do with charge. It's more like if you run a test. Are you pregnant or are you not? Are you positive or are you negative? So in this case, for this means if you're positive, you stay purple through this whole procedure. If you're a negative, you don't stay purple. Instead, you get stained pink. Still with me? We're staining the cells, so what type of dyes are these? Basic or acidic? Basic. And they have what charge to them? Basic dyes. Positive. So that's why they're attracted to these negatively charged cells. All of them are negatively charged, right? But when we say gram, right, we're talking about, does it stay purple, positive, or negative, ends up being stained pink. So we're going to start with positive. And so this is a diagram representing the cell wall of gram-positive bacteria. So, or you could say the cell envelope, which is made up of the cell membrane and the cell wall. So here's the membrane, those phospholipids we're used to seeing, right? If you click, ID molecule, it gives you the little key. That's a phospholipid, right? That's teichoic acid. The membrane proteins, peptidoglycan, crystal violet, the different dyes that we're going to use. So, and here's a little script down here, but I'm going to kind of add my own as well. So the first step of the Gram stain is to add the primary stain, crystal violet, which is a purple stain. So we're going to add that. Guess where it goes? Inside the cell, right? The next step is we add grams iodine, or just straight up iodine solution. This goes into the cell as well. When it does that, though, it complexes with the crystal violet to form what we refer to as a di-iodine complex, or a crystal violet iodine complex. It makes it a much larger molecule than crystal violet and iodine by themselves. This is an important aspect of the staining procedure. The fact that we end up with a bigger molecule, dye molecule on the inside. The next step is decolorization. Some people would use acetone, but ladies, we all know, right? That stuff is really harsh and damaging to mucous membranes. Ethanol works just as good, right? Alcohol. So we use that instead in the laboratory nowadays. Watch what happens, right? So the D is for the decolorizer, right? The ethanol. Whoop! What happened? It shrunk. What shrunk? No, that's not the membrane. What's this? That's the cell wall. And what is that made out of? Peptidoglycan. The peptidoglycan shrunk. Now, that cell wall, is it like a wall like these walls? Nothing can get through? No, that wall is more like chicken wire, right? It's really pretty stable, but stuff can get in and out, little stuff, which is important for the cell, right? Stuff's got to get in and out. So it's structurally very rigid, right? But there's big gaps in it. But what just happened to those gaps? They shrank, right? They got smaller. So do you think that crystal violet iodine is going to be able to get back out? No, it gets trapped in at this step. So then the last step, see it's trapped, is adding the safranin. That also goes in. It's small, right? This is a red dye, but we already stained them purple. Which are they going to look like? Purple or red? Purple. The best way to get people to understand this is why we do purple first, is laundry. When I was in charge of laundry once, I got a red sock in with the whites. My dad had pink underwear for a couple weeks. He was a cool dad, though, so he wasn't too mad at me. My husband, on the other hand, don't tell anybody. Black underwear. I love this man. Do I ever have to worry about screwing up laundry? No, you're not going to notice, right? Something that's black, if red gets on it, it's still going to look black. It's awesome, right? We don't sort clothes. We put it all in the cold cycle, all together. Cold is actually what you're supposed to wash your clothes in. All the stuff you buy nowadays is formulated for cold water. If you actually put it in hot water, it's not as effective at cleaning your clothes. So save your electric bill. Cold, throw it all in. If you're worried, you can buy those little sheets that soak up color. Shout or somebody makes them. Make sense? Okay, so they're going to be what color? Purple, so they're gram positive. So obviously it's different for negatives, right? So there they are, they're purple. Let's do gram negatives. Oh, wow, it's already different, huh? Wow, what is all this stuff? We've got new stuff up here, huh? What is this? What is this? Right? So what is that? Ooh, lipopolysaccharide. That's neat. And that looks like what? A membrane. But it's not the cell membrane, it's what we call the outer membrane. So here's the cell membrane, or the inner membrane. There's our good friend peptidoglycan, and what do you notice in comparison to the gram-positives? This layer is what? Thicker or thinner? It's thinner, right? And we've got this outer membrane. So do you think it's different things are going to happen? Sure, because we end up with a different color, right? So something different's going to happen at some point, right? So first step is always crystal violet, though. So it's going to go in, right? They're going to be purple. We're going to add the iodine. It's going to attach. Make it bigger. They're still going to be purple. Okay, decolorize. We're going to get shrinkage? Yeah. But what else is going to happen? Watch. Decolorize. We lost a whole layer. What layer was that? Outer membrane. What are membranes made out of? Other than that funky lipopolysaccharide. What's this stuff? Phospholipids. Lipids. Fats, right? What does ethanol do to fats? It dissolves them. Guess what's in your hand sanitizer? Ethanol. Alcohol. Why do you think your hands end up getting dried out? Dissolves away your lipids, your moisture, your fat. But it also does that to gram-negative bacteria like E. coli that might be on your hands. And kills them. But that's also why they try and put moisturizers in those things to get you to keep using it. because, you know, can't stop taking away your lipids. Can't distinguish between your lipids and E. coli's lipids. So do you think that crystal violet iodine is going to stay in there? Nah, not enough. Not enough layers here, right? Not enough shrinkage. They escape. Gone. So if we were to stop here and look at these cells under the microscope, would they have any color to them? No. Do we want to see them, though? Oh, of course. We always want to see them. So what do we got to do again? We got to stain them. But we got to pick a different color, right? So we know this happened. So now it's saffronin, which is actually a red dye. But these guys are going to appear pink because they're not going to absorb a lot of the red dye because of the damage that we cause when we decolorize them. So they're going to be pink. But now we know why that's important, right? That tells us about the cell envelope structure. How many membranes does it have? How thick is its peptidoglycan? And we'll talk about the significance, medical significance, right, when we get to our next topic. So you know me. I love tables, right? See if you can fill it in, right? See if you can, you know, remember what happens at each step and why, right? And then the answers are here, but I'm not going to give them to you yet. That's homework. This is a little quiz. You ready? How do you get that ball out that hole? How do you get that ball out of that hole? Take the pegs out. Take these little sticks out, right? Because they're not going to let it go through. This is actually an intelligence quiz, by the way, right? And clearly the ball went in and then the sticks got put in. So how many of you guys have seen this toy? My mom actually still has ours. I was tempted to steal it when I visited. Right? No. So this is a common little kid's toy. It has little blocks, you see them on the inside, but they have these particular shapes to them. So the block only fits in the shape that it matches with. But notice there are two halves. There's actually a little handle here at the top and the bottom. The two halves come apart, so you can dump all the blocks out and then put them back in. Most little kids don't know that. So you give them all the blocks, and then they stick them in, right? So it's all filled up like this, and of course they want to do it again, right? So what do? They stick their little hand in one of these holes, and they grab a block. Then they start screaming, right? Because can they get their hand out? No, that block barely fit in there by itself, never mind surrounded by a little hand. So once you finally convince them to let go of the block, right, and take their little hand out, then you can show them how to do it properly. How is this related to the gram stain, though? How's that related to the gram stain? What the kids do? What do we do in the gram stain? What's the first step? Put crystal violet, right? Where does it go? Inside. What's the next step? You put iodine in. And what does the iodine do?
The iodine makes the thing bigger.
Makes it bigger. It attaches to the crystal violet, right? So the same thing here. Block goes in, little hand goes in. Little hand grabs block. Cannot get back out because it's too big, right? But in the gram stain, can you imagine that if this happened to the kid? With the gram stain, crystal violet goes in, iodine goes in, right? Then what happens to the hole when you decolorize? It gets smaller. Can you imagine a kid putting his hand in, grabbing the block, and the hole got smaller? You'd seriously have somebody screaming. It'd be over with. Yeah. It would. Yes, it can be. It can be. So, For homework, see if you can, right? Start reading, at least watch those animations. The next PowerPoint is up.